lecture, we are finishing up Chapter 15 from Voet and Pratt, where we will discuss the metabolism of other sugars and the pentose phosphate pathway. In class, we will then apply the information from this lecture in our activity S31 from the workbook to learn how to identify where other sugars enter into glycolysis, predict the biochemical route followed by glucose given various cellular needs, identify which carbons from glucose are converted to CO2 during the PPP, identify which biochemical processes in which NADPH is an important cofactor, and also gain some additional practice in transferring knowledge from one context to another. Specifically, taking the rules and patterns we learned about in Chapter 14, Introduction to Metabolism, and applying those to a more specific situation, such as the pentose phosphate pathway. So, in our last unit, we studied how glucose is converted to pyruvate in glycolysis. While glucose is our primary sugar used by the cell to make pyruvate, we can metabolize other hexoses, or 6-carbon sugars. Specifically, galactose, mannose, and fructose. As seen here, galactose intersects glycolysis at G6P, and mannose intersects at F6P. Fructose, however, intersects glycolysis at F6P if it is being metabolized in the muscle, or at GAP if it is being metabolized in the liver. Looking more closely, galactose is converted to G6P in a 4-step process where it is first phosphorylated, transferred to an activated UDP molecule, isomerized to glucose, and then rephosphorylated through an exchange reaction with a molecule of UTP. Patients who lack the enzyme that govern the second step of this process, galactose 1-phosphate uridyl transferase, due to an autosomal recessive genetic abnormality, will have a dangerous buildup of galactose 1-phosphate, which causes mental retardation, liver damage, all-around failure to thrive, and is potentially fatal. This disorder, which is significantly more prevalent in Irish populations than others, is extremely fatal in newborns. So now nearly all newborns are tested for galactosemia at birth. However, the most common treatment for this disorder is to simply eliminate galactose and therefore lactose from the diet. As I mentioned previously, fructose is metabolized differently based on whether it is being metabolized in the liver or in the muscle. In the muscle, it is simply phosphorylated by hexokinase to make F6P. However, the liver isoform of hexokinase is much more specific and does not recognize fructose as a substrate. Therefore, it is first phosphorylated by fructokinase to make F1P and is then cleaved to form DHAP and glyceraldehyde. DHAP can then be directly isomerized into G3P, continuing its path through glycolysis, where the glyceraldehyde is phosphorylated using ATP and a kinase to make G3P. Metabolism of fructose in the liver therefore bypasses one of the most important glycolysis regulation steps we discussed last week, PFK1. Because we bypass this major point of control, all of the fructose that is metabolized by the liver is immediately converted to pyruvate. If ATP needs are good and the citric acid cycle is turned off, that excess pyruvate will eventually be used to make fatty acids, causes a buildup of fat around the liver. Excess fructose consumption through sweeteners such as high fructose corn syrup in sodas and other American food staples are therefore leading causes of fatty liver disease and obesity, which can lead to cirrhosis of the liver and/or liver cancer. The pentose phosphate pathway is utilized by the cell to develop NADPH, another reducing agent that is utilized in biosynthesis of macromolecules, versus NADH, which is used to help synthesize ATP during the breakdown of macromolecules. The PPP starts with G6P from glycolysis and eventually generates NADPH, CO2, F6P, and GAP. Because F6P and GAP are also metabolites in glycolysis, these pentose phosphate pathway products can still be utilized by glycolysis or gluconeogenesis. The pentose phosphate pathway can be broken down into 3 stages. In stage 1, we have a series of 3 oxidation reactions to yield NADPH and ribulose 5-phosphate. In stage 2, we have an isomerization and epimerization. In stage 3, we have a series of carbon-carbon bond cleavages and carbon-carbon bond forming reactions to make F6P and GAP. While stages 2 and 3 are freely reversible, stage 1 only operates in the forward direction. We will see why this directionality is important in our in-class activity. In the first step, G6P from glycolysis is oxidized by glucose 6-phosphate dehydrogenase to make our first molecule of NADPH and a gamma-lactone intermediate. This enzyme is strongly inhibited by NADPH, meaning if the cellular needs of NADPH are satisfied, this enzyme will be turned off. In the second step, we have hydrolysis of the lactone metabolite to make 6-phosphogluconate, which is then further reduced to create our second NADPH molecule, followed by a decarboxylation step to release CO2. This CO2 coming from carbon 1 of glucose, creating ribulose 5-phosphate. This completes stage 1 of the pentose phosphate pathway and creates 2 molecules of NADPH for each molecule of G6P that enters the pentose phosphate pathway. In stage 2, isomerization and epimerization converts ribulose 5-phosphate to either ribose 5-phosphate or xylulose 5-phosphate, depending on the needs of the cell. In cells that are actively undergoing replication, ribose 5-phosphate is in higher demand as it is utilized in nucleotide synthesis. Otherwise, xylulose 5-phosphate is created twice as much due to the third stage of the pentose phosphate pathway. In stage 3, xylulose 5-phosphate and ribose 5-phosphate are consumed by a transketolase to make G3P and S7P. G3P can be directly used by glycolysis, while SU7P is used in step 7 along with a molecule of G3P to make E4P and F6P. Again, F6P can be used directly by glycolysis, while E4P and a second molecule of xylulose 5P are converted to G3P and F6P, both of which can be used in glycolysis. In summary, the pentose phosphate pathway is utilized to create NADPH and R5P. If R5P is not needed for nucleotide synthesis, it can then be converted to the glycotic intermediates of GAP, F6P. The flux of the PPP is controlled by the rate of G6P dehydrogenase in the first step of the pentose phosphate pathway and is inhibited by NADPH or activated NADP+. We can then further control flux through this pathway through hormonal control of the cellular expression of G6P dehydrogenase, which is explained in box 15.4 of your textbook. Patients who are deficient in the enzyme G6PDH will have a limited supply of reduced glutathione, which is an essential antioxidant to help maintain healthy erythrocyte function. This enzyme deficiency is more prevalent in African, Asian, and Mediterranean population and causes hemolytic anemia. But as seen in sickle cell anemia, deficiencies in this enzyme seems to lead to malarial resistance, which is what causes us to see a higher proportion of this genetic abnormality in African populations than others. All right, I will see you guys in class.